So today we're going to talk about wearable device data. And as Hugo alluded to this morning, we're now living in an age where we have an increasing number of wearable devices. So these include things like continuous glucose monitors, smartphone food applications to record meals, other things to measure blood pressure, Fitbit, all kinds of different types of data. And so these wearable devices produce these time series data, and the global question, which is very sort of vaguely defined, is starting from these time series, how can we best summarize and capture relevant information, particularly when we have multiple wearable devices? So to answer that question, we a few years ago, with funding from the PHRT, this was a collaboration between Phillips when I was a postdoc at the EPFL and Tinhai Kole, so I'm working with him now in Geneva. And the idea was to measure multiple wearable devices in 25 healthy participants over a 2-week period. So these devices included, we can see here, this Freestyle Pro. So this measures your interstitial glucose, something you wear on your arm. It measures every 15 minutes for 2 weeks. We were also using an app called My Circadian Clock, so participants record their meals when they're eating it, and also free text annotation. And then we also used a third device called the ActiHeart 5, so this measures physical activity with an accelerometer, but also heart rate and heart rate variability. And these are using electrodes, so the signal quality is typically regarded as being high, but the problem is with electrodes, often you're getting missing data. So the best way of sort of approaching this data is just to start with some basic visualizations. So, so these 3 columns represent 3 different participants over 3 different days. And so if we look at the top row, we can see the glucose data in the blue, and we can see the meal recorded with the app recorded as vertical gray lines. So for some participants, it sort of looks a bit like as you might imagine it. So here we can see that, okay, there's a meal for breakfast, and then it seems like there's a large, predictable glucose spike afterwards. But what you need to know is that kind of the real data, it actually often doesn't look like this. So in the second example, we can see someone who, after their meals, it looks like these postprandial glucose spikes look pretty small, and they look barely larger than the fluctuations in glucose that are occurring in between meals. We can also say, okay, maybe it looks like there are possibly some oscillations going on between meals, although it's difficult to say. And also, we also notice these fluctuations even during the night. So the glucose is never at this perfect steady state. It's always kind of fluctuating. And the third thing we noticed just through data visualization is that sometimes it looks like there might be glucose spikes even in the absence of a meal. So here on the top, we see the glucose data, and there's a big glucose spike even when there doesn't seem to be a meal preceding it. And if we look at the activity data, and the heart rate data. So this was during a period of physical activity. It's known that the physical activity can provoke glucose spikes. But the question is a bit more systematic, is can we use the activity data, the heart rate data, the heart rate variability data to help us understand glucose dynamics? So the overall aim is to try and combine these signals together. We're first going to start with looking at food and drinks on the glucose levels. Then we're going to look at the impact of the circadian clock. And as the final part, we're going to try and combine these additional signals into predicting glucose. So our modeling approach in some ways is quite simplified and even cartoony, I describe it, because the underlying assumption is that our glucose is basically a homeostatic mechanism where we can see on the right with this diagram of it's bad to have sort of too low glucose and it's bad to have too high glucose. So when we have a meal, marked again in this vertical line, glucose will go up, but then there's a range of homeostatic mechanisms that will normally try and bring a glucose back to steady state. So the way that we can try and model this mathematically is by a sort of a dynamical system which is receiving external inputs. So we model this as a very simplified dynamical system where so the system receives inputs from the meals, which cause a glucose spike, and then the glucose will return back to baseline with some kind of characteristic timescale that we refer to as a response half-life. But the important thing is that the response height might not be the same for every meal. So this needs to be a learnable parameter. So every time you're having a meal, you somehow need to learn this response height. Although within our mathematical framework, the overall half-life is as a kind of a global dynamic parameter. Importantly as well, we model this with a negative feedback loop, and this allows us that in some case, for certain parameters, it means that if you have a glucose spike, you can also get some overshooting, which is something that we sometimes see physiologically. And the other important point is that, so we can define a mathematical model that's receiving these perturbations, but we don't know any of these parameters before we start looking at data. So the goal is going to be to try to infer some of these parameters from the data. So the first approach was just to use the meals to try and predict glucose. So the glucose, here we see over the 2-week period, an example of a glucose trace, which is shown in blue. And then the meals are shown as these, again, as these vertical gray lines. And what the orange model is this is a model that's using just exclusively the meals to try and predict the glucose. So the model is, during the inference, is adjusting the meal heights to try and maximize the fit. And the overall fit is okay, so the correlation between the orange and the blue is about 0.68. But there's a problem, and the problem is that if we look into detail, is that if when we only use the meals, there's a model residual is the difference between our model in orange and the data in blue. And the model residual shows a systematic error in the sense that there's a systematic 24-hour fluctuation going on in the sort of in this model residual. So our approach to resolving this problem was relatively simple. We simply add a 24-hour function to our mathematical model. So we use a 24-hour cosine function. So this has 3 parameters. It has a kind of a peak time, a baseline, and then an amplitude. So we can add this to our model, which we see in this black line. And then when we include an underlying 24-hour function plus the meals, the overall model fit here is increasing to about 0.76. So the first, if we revisit this question of this dynamic response half-life, if we then apply this approach to kind of real participant data, we can see that the model is able to identify individuals with either fast or slow dynamics. So here are 2 individuals where we see the data over 4 different days, and we see that for this ID that's identified fast dynamics, you can just see how rapid the glucose responses are, meaning that after each time that there's a glucose spike, the return to baseline is very fast. Whereas with the slow dynamics, you can see visually that after there's a glucose input, that it's taking much longer to return to baseline. So if we look over our population, we found that the individuals typically who had longer half-lives, meaning that they take longer to return to baseline, typically had larger meal heights, so they have larger postprandial glucose spikes. So there are several biological explanations for that. So it could be that the individuals with these slow dynamics somehow either have insulin resistance or they have pancreatic beta cell dysfunction. So these are 2 different mechanisms that could cause to sort of perturbed homeostasis. So one idea we've had for the future that would be really interesting is that there are ways of trying to estimate this by using these standardized meals, so either mixed meals or these oral glucose tolerance tests where you give individuals 75 grams of glucose. But you need to measure glucose, insulin, and C-peptide for some of these models to work. So you have the logistical complications of doing that. The next thing, if we return to these rhythms, so the way we're learning these parameters is within a Bayesian inference paradigm. And what that means, and we're using the MCMC, so Markov chain Monte Carlo, to estimate these parameters. So if we go back to this amplitude parameter, here we can see that for one individual, these box and whisker plots represent the uncertainty on this amplitude parameter for one participant. And what's interesting about this, the kind of MCMC and the Bayesian approach is that now that we have these confidence intervals, we can compare different individuals. So here we see an individual with very high amplitude, and we see the confidence intervals are quite tight. And in other cases, this amplitude seems to be very, very low. So here are the visualizations of these two individuals. So over the 2 weeks, we can see that there does appear to be this underlying 24-hour structure in the glucose data, whereas for this other ID, so this is for the individual with a very low amplitude parameter, this thing is basically not existing. So it's you could the way that we describe this is a bit sort of phenomenological in the paper, but one of the things we're interested in is applying this in a disease context. So we currently have a project we're working on at the moment on gestational diabetes. And when we calculate these parameters in a post-gestational diabetes group and a healthy control group, we do seem to see systematic differences between these groups. So we're preparing that in a manuscript that we'll hopefully submit within the next few months. And the final part was to this idea of combining the different signals to help us predict glucose levels. So here's an example, which is a very extreme example of someone over their recording period, where you can see that periods of intense physical activity, so in this case it was CrossFit, they seem to coincide with some of the largest glucose spikes over the entire study period. So when we try to integrate these signals onto the glucose levels, so this is using a Kalman filter approach, we found that the physical activity generally had a negative effect on the glucose levels, while heart rate seemed to have a slightly positive effect, while HRV, which is heart rate variability, has an overall relatively neutral effect. This was a bit of a sad result in a way, because we went through a lot of effort to try and capture this HRV data, which is quite complicated, and at the end of the day, it doesn't necessarily seem the most important thing for predicting glucose. So here we can see in this bar plot, this is the light orange is representing how much of the variance do we capture in the glucose model when we use only meals and glucose, and the dark red is how much variance we capture when we add the activity, heart rate, and HRV. And the result is somewhat difficult to explain, because for some individuals, it really does seem that you get a nice boost in how much glucose variability you can explain. So for some individuals, it seems to go up to about 15%. But what's a bit complicated is that they seem to be very heterogeneous. So there seem to be many individuals where the relative boost you get in adding these three signals is pretty low. Whereas, as I said, there are some examples where you can sort of partially explain glucose spikes when you incorporate these data. So the question is, should you add to these signals? Is a bit, well, maybe in some cases it's useful, but in many cases it might not be so useful. So just to sum up with a few of the key lessons, I think what's really interesting about this wearable data is that it allows us to see dynamic responses. So in real time, as opposed to something like a fasting glucose measurement, it allows us to see how the body is responding dynamically according to stressors, and also allows us to see these 24-hour rhythms, because we can record over a long period of time. Another interesting advantage of wearables in this case is that it's relatively easy to set up the experiment. So there were two of us to do the setting up all of the data collection, and it took us 2 days to give all of the participants all of the wearable devices. But one of the difficulties in our approach is that, as we discussed, the model is based on these glucose perturbations, and this is not really taking into account precise macronutrient data. So now we're using a new app called MyFoodRepo, which does include some detailed macronutrient data, but it's actually somewhat difficult to include in our model, because it becomes very complex. For example, if you're thinking about how fiber might influence the shape of these glucose responses, the modeling task becomes a bit more complex. So it's a bit on the to-do list for the moment. And the kind of the final main thing is that so the MCMC allows us to compare different participants, but a more interesting use case would be to say, okay, we have time point 1 and we have a time point 2. This is across an intervention, and to be able to see the differences. For example, we have an ongoing study at the moment on intermittent fasting. So we're hoping to recalculate all of these parameters before intermittent fasting and after intermittent fasting to see what's happening. So with that, I'd like to thank collaborators, Tinhai Kole and Felix Neff, and thank for all the funding from PHRT and the rest of the team. So thanks for listening, and if you have any other questions, I'm also presenting a poster.